Bismillahirrahmanirrahim. Lecture number 4 about the polynucleotides. There are two types of polynucleotides, DNA and RNA, deoxyribonucleic acid as well as the ribonucleic acid. This is the lecture 4 about the polynucleotides. These polynucleotides, they are basically the phosphodiester bond. They have the phosphodiester bond. These nucleotides, they link through the phosphodiester bond as well as their phosphate groups are also attached at the 3' hydroxyl of the pentose sugars, and also the second phosphate group attached at the 5' side, and the phosphodiester bond, which is the directional at the 3' to 5' direction, it forms the backbone of RNA and DNA. RNA and DNA, they are the types of nucleic acids, ribonucleic acid as well as the deoxyribonucleic acids. Hydrolysis of the phosphodiester bonds of DNA, it occurs only over a long period of time. It is due to the absence of the nucleophilic species in the DNA, as oxygen, it is at the 2' position. The oxygen, there is a deficiency of the oxygen, and oxygen act as a nucleophile, or it also, in the presence of this oxygen, it also reduce the strength of the nucleic acid. DNA contain, it is the deoxyribonucleic acid. There is a deficiency of oxygen, so it is more stable as compared to RNA. This is the representation how the phosphodiester bond is direction, that is in the 3' to 5' direction. All of the nucleotides, they are joined by means of this phosphodiester bond, and this phosphodiester bond, it is directional in the 3' to 5' direction. Along with, there is a phosphate group present at the 5' position, and also the hydroxyl group present at the 3' position, while the nitrogenous bases, they are the perpendicular to this phosphodiester bond. And again, this is another representation in which the nitrogenous bases, they are attached. These representations show the nitrogenous bases through the linking through the phosphate group. In the post-transcriptional modification, the polynucleotides, there are the formation of the additional nucleotides such as the pseudouridine, as in case of tRNA. In the pseudouridine, there is a linkage between the carbon number 5 of the uracil with the D-ribose, carbon number the carbon of the ribose sugar. Here there is a carbon to carbon bond instead of the beta and glycosidic bond, as the sugar, it is attached to the nitrogen of the nitrogenous bases, but in the pseudouridine there is a carbon to carbon linkage instead of the beta and glycosidic linkage. And by this way, a nucleotide pseudouridylic acid, it is formed, and the rearrangement of uridine monophosphate, it also give rise to the TMP. By the methylation of UMP, it gives rise to the TMP, but in this case, this thymidine monophosphate, it contains the ribose sugar rather than deoxyribose. This is the polymer of nucleotides. Here the formation of the phosphodiester bond, here the 3' to 5' direction, it is the directional, and all of these, they are the nitrogenous bases in the DNA, because DNA contains the thymine. This is the DNA, one of the strand of the DNA. According to the structure of DNA, it is defined by both Chargaff as well as the Watson and Crick model. According to Chargaff, there is a DNA molecule in which the concentration of deoxy adenosine nucleotides it is equal to the thymidine nucleotides, mean A is equal to T. Similarly, the concentration of deoxyguanosine nucleotide it is also equal to the deoxycytidine, mean G is equal to C. Another model proposed by the Watson and Crick, this model shows that there is hydrogen bonds between the purine and pyrimidine. Along with this hydrogen bond, there are the van der Waals as well as the hydrophobic interaction between the stacked adjacent base pair. And the helix, it is a helical structure, and this is the right-handed helix, and the base residues, they are formed in the spiral in the clockwise direction. Here there is a restriction and which is imposed by the rotation about the phosphodiester bond. So the phosphodiester bond it is rigid, and there are the predominant tautomerism. As in the keto-enol tautomerism, the predominant keto and enol tautomerism, they are the predominant. Amino and amino tautomerism, it is the predominant. Similarly, the keto and enol, the keto tautomerism, it is predominant tautomerism. A singleton of bDNA, different forms of DNA in which the bDNA it is the usual Watson and Crick model, and it's about the long axis of the molecule contains about the 10 base pair, and the distance spanned by one turn of bDNA it is about the 3.4 nanometer, mean 34 angstrom, and the helical width is about 20 angstrom or 2 nanometer. This the 20 angstrom its width, and 34 angstrom it's one turn of helical structure. There is a formation of the major group and along with the minor group, and proteins they are embedded in both groups. The opposite strand it is known as the coding strand because it matches the sequence of the RNA transcript. So the template strand, sometimes known as the non-coding strand, and its opposite strand it is known as the coding strand.